many of you know, today's lecture is in honor. It's our annual tribute to Dr. Vince Cristofalo, who really was a pioneer in aging research and was the founder, actually, of the Center for the Study of Aging, which is now turned into the Institute on Aging here at Penn. He actually did seminal work in standardizing and learning about the aging of cells. And this classic work has been used by scientists and has had broad implications for many years since. While he was focused on cellular aging, he was also really focused on linking basic aging research so that it was accessible to all aging-related research and the study of gerontology, and so had a very sort of broad perspective on his work, and was known as a excellent mentor for students, trainees, and junior faculty. And we've had just a tremendous list of awardees who've done this talk in the past, having gone to many of these over the last 10 years. It's been just a sort of honor roll list of wonderful speakers, and I think Dr. Cristofalo would be especially proud of that list, as well as today's speaker.
David walks on it. David walks on speaker.
For today's talk, I have the great pleasure of introducing Dr. Jeffrey Kaye, who traveled here all the way through the internet from Oregon remarkably quickly. Dr. Kaye is the professor of neurology, the Leighton Professor of Neurology and Biomedical Engineering at Oregon Health and Science University. He directs their Aging and Alzheimer's Disease Research Center, the Oregon Center of Aging and Technology, and their Roybal Center for Care Support Translational Research, advantaged by integrating technology. Dr. Kaye did his medical school at New York Medical Center and neurology and movement disorders fellowship at Boston University. And really, there's not an area within the aging and Alzheimer's disease research and dementia field that Dr. Kaye has not been involved in. He's done work on observational studies, on studies that involve clinical trials. He's done neuroimaging work. He's done behavioral and cognitive studies. But probably, I think what he's most famous for is he really has been, I think, an international intellectual leader in the use of technology to better monitor in more ecologically valid contexts changes that occur in the context of aging and early neurodegenerative disease. And this work has spanned, I think, an incredible scope, but in particular, work that's done in an individual's home, which is potentially has incredible implications for the reach of our ability to monitor patients in the context of trials, but also in the clinical context as well. And so it is really my great pleasure to introduce Dr. Kaye and to hear about his way of the new vision for looking at Alzheimer's disease in the talk today. Thank you, Dr. Kaye.
Well, thank you very much. We're gonna.
20 years, we had a center called the Leighton Center on Aging that was named after the Leighton family. It's a rather unusual name, so maybe it occurs in many places. But I never knew when we set up our Leighton Center if it was connected at all with what you do. And it's an easy yes or no answer. But we had a Leighton Center on Aging for about 20 years.
Well, knowing the Leighton family in Oregon, I doubt it, but everything is 6 degrees of separation, so who knows? In any event, I want, first of all, thank you to the organizers for having me virtually. I very much was looking forward to coming to Philadelphia. I grew up at the Jersey Shore, a term that's probably more recognized by people back east than out here. And so I was actually looking forward to coming east, but obviously that was not possible. I hope, in fact, that I will have the opportunity to come subsequently in the near future. I also actually want to thank upfront Ebony Fenderson for her help in facilitating getting this organized and sending me the links and all the things that are needed to get this going. So with that, I want to just start out by which here I'm going to start. I call this the Gloom and Doom slide. It's the almost the catechism of presentations about Alzheimer's disease that we talk about the statistics, and they are quite daunting. So we all have this number from the Alzheimer's Association of 6 million people living with Alzheimer's disease. The fact that it's has a high mortality, 1 in 3 seniors die with Alzheimer's or related dementia. During this time of the pandemic, this has been heightened with the brunt of people in the pandemic being people with dementia being affected. This has also been seen in terms of populations or communities of color, which are seen to have difficulty or discrimination when they seek health care. And this is also seen not unique to general health care, but specifically with regard to dementia. The challenges in general are huge for not just the person directly affected or with the disease, but the caregiving population, people who are generally family members, unpaid care workers, and the cost of this is huge, over approximately 257 billion estimated this past year. And then ultimately, the cost to the nation is tremendous, with the projections going out to 2050 of over $1 trillion, which is, when we start talking trillions of dollars, we're talking about the size of our infrastructure packages, for example. So there's huge implications, of course, about this disease. Having said that, one of the incredibly sad implications about this disease, or Alzheimer's disease, is that we really don't have meaningful treatments. And we know that there's been a large number of trials over the years. So this figure that you see shows that what's a pretty big funnel at the top. We have lots of candidates, but a only tiny trickle come out of the bottom of the funnel. And in fact, those that come out of the bottom, that so far have come out of the bottom of the funnel, are really not very effective. There's only a handful. They're really symptomatic treatments. And there hasn't been really a new mechanism or treatment since approximately 2003. Now, this is not for lack of trying. There's over 400 failed trials, at least. Currently, this is updated from Jeff Cummings' review that there are about 121 therapies in clinical trials in the past year. The colorful diagram shows that there are many, many different mechanisms being looked at. About 80% are so-called disease modifying. A smaller percentage, 20%, are symptomatic treatments, which are a good symptomatic treatment would be very welcome as well. But ultimately, this is still a work in progress, and it's an expensive work in progress. The cost of bringing a drug to market is very high. It's over $2.5 billion. These are estimates. They're actually hard to get these numbers to be well secured. So why are there no meaningful treatments? And I would argue there's just a couple main reasons. One is we actually don't know what the causes of Alzheimer's disease are. And I know some people may say, oh, wait a minute, what do you mean by that? Well, what I mean by that from a drug development point of view is what is the target? So we've been shooting arrows at a lot of targets, and I think the previous diagram reflects this, that we are struggling to know really what is the right target. And so there's right now a lot of movement to look at different targets, and that's not perhaps surprising. The other piece, though, is that I would argue that we don't really know what success looks like. And what I mean by that is what would a treatment actually show if it had the ability to change the disease course meaningfully? And I think, here, as I'll show in a moment or discuss more in detail, there are many potential outcome measures. Right now, in the trials world, we use basically cognitive test results of various kinds, and perhaps some functional surrogate measures or self-report measures to suggest that there's some change that's been occurring over some period of time. But unfortunately, this really, I think, has led to us shooting ourselves in the foot in a way that we may have we may be predicting that we're going to lead to failure, because in effect, right now, we're looking for small effects because of the kinds of endpoints that we're measuring that are that result in expansive, long, and expensive trials. And so this is really the kind of underlying theme of what I'm going to talk about this afternoon. So let's unpack that a little bit. So right now, the tools we use for detecting change in trials are really a couple of mainstays. One is cognitive tests, and these are the long-standing legacy test is the ADAS-Cog, and then various confirmations of that measure. But in general, they're all have the same problem. They, as you see here from ADNI data going from left to right, normals, people with MCI, and people with early Alzheimer's disease, there's tremendous heterogeneity, both within an individual as well as between individuals. And so that clearly is a problem of assessment. And this is true also for the mainstay of functional measure or integrated functional measure, the CDR, the Clinical Dementia Rating Scale. This is just some data shown from a South Korean longitudinal study, subjective cognitive impairment on the top, amnestic MCI in the middle, and dementia in the and Alzheimer's dementia in the bottom. Again, showing the tremendous heterogeneity intra-individually and across subjects. But these have become the canonical measures that have been used repeatedly in trials, and they make for very difficult trial conduct. To look at a little bit, just a little more detail at the CDR itself, this is the famous grid, the sort of 5x6 grid. If you focus though on early stage dementia, prodromal, MCI stages, you're really talking about just this upper quadrant, because the bottom rows are really functional measures, which if they're impaired, the person is by definition demented. So this grouping really then makes the CDR into essentially a 6-box change potential, and that really doesn't make it that much more sensitive. Further, there are very few studies that have actually shown that raters are able to accurately rate CDR changes in this early stage, the CDR 0.5, 0 to CDR 0.5. This is an older study done by the ADCS in a clinical trial training a bunch of raters, over 80 raters in a room, to get them all standardized on the CDR. And the bottom line is, if you look at the agreement among raters, and when we talk about experienced raters, this is people who have done other clinical trials, that were in the room looking at the same case studies, videos, and then rating the CDRs, you basically could have flipped a coin in terms of the agreement of CDR 0 versus CDR 0.5. Nevertheless, this, if you go into clinicaltrials.gov, most or many trials are still using the CDR sum of the boxes as a primary outcome measure. Then finally, just to maybe overemphasize this point, but I wanted to also make this comment that as a clinician, so I see patients as well, and when we look at thinking that we are really good at knowing how people are changing when we examine them, see them, interview them, the clinical impression of change, which was an older measure, sort of a composite measure of whether somebody is getting better or not. This was a study done years ago where the interviews were videotaped, literally with videotape. We don't have tape anymore. But the tapes were then sent around to some very distinguished neurologists around the country, and they were asked to rate these individuals as they went through the trial. And we didn't tell the neurologists that some of the tapes were reversed. So by the way, of course, the drug didn't work, people got worse, but the raters didn't know that. And so if we reverse the tapes, they would be getting better because they didn't decline in that way artificially. The bottom line is that about 63% didn't actually pick up on the fact that they were getting better. And I also should point out that one of the wonderful authors here was Penn's own Chris Clark, who was a near and dear colleague of mine and many others, I'm sure, at Penn. So this, I think, for all of us, should be taken to heart, but we still press on. And this just then makes the whole idea of how you understand whether there's a meaningful change happening or not more complicated. And the implications for the sample sizes in particular are huge. And so just to demonstrate that, Ron Brookmeyer has done some modeling using the Mayo Clinic data, so much more of a population-based study, looking at going from normal to amyloidosis to MCI to dementia through these different states, and projecting what those rates of change are, and then the sample size needed in a trial. If you then consider more recently, Bill Jagus and Susan Landau looked at using ADNI data, looking at the time it takes from going from a fluvedapir positive scan to MCI. You realize that there's, first of all, tremendous variability, even though this is a smoothed curve because it's a modeled curve, between individuals who progress and those who don't. So again, getting back to that heterogeneity problem. But that the time from a positive scan, amyloid scan, or amyloidosis to MCI is about 14 years. So really, you have to have a long observation period or lots of people to see people who are changing faster than others. And then if you then project these facts onto sample sizes needed, these are just different probabilities based on the effect sizes that you might desire in young or older populations. And just to the bottom line is, or columns are, that you need thousands of individuals in these trials to find an effect of a treatment using actually the transition to a clinically meaningful state like MCI. So we have a critical need for having a better way of understanding whether people are meaningfully changing. The FDA has recognized this and basically has said that we need an outcome measure that will provide an assessment of meaningful cognitive function, particularly in this early prodromal kind of stage. And they encourage the development of novel approaches to the integrated evaluation of subtle early AD or pre-dementia, functional deficits, that arise from early cognitive impairment. And so this is, I think, fundamentally recognized as a challenge. So how can we challenge that? How can we change that? Change the way we see change itself. And this really brings me to the digital measures idea that, better measures or measures that give you an outlook that is more meaningful is what's needed. This has been a centuries-long process that Leonardo da Vinci wrote about, saying that, measurements of man are distributed in an interesting manner. 4 cubits make a man, a pace is 4 cubits, a man is 24 palms. And these measurements are in his building. So the way we are move about, the way we exist in our environments are reflected by our environments, this concept of ecological validity. And so the idea that we can have some measurements that really are better reflections of what we're doing in our real world is the key. So there are many of these so-called digital biomarkers or measures. There are hundreds of wearables that are not just wrist worns, but body worn, embedded in various clothing and shoes, and there's just a host of these available. These are often paired or even freestanding for other purposes with devices such as phones or tablets and apps. There are, better yet, to my thinking, are more passive sensors and devices that collect information in the background so that one doesn't burden individuals with extra activity. And then there's the integration of multiple channels of information that can be derived from these various devices and sensors. But what we know is we live in a sea of these devices and sensors. Where's the evidence of whether these things work and can deliver the information that we're looking for? And I'll just summarize very quickly a lot of the information here that I would say that this field is still actually, relative to other fields, quite small. If you compare it, for example, to proteomics or any other of or wet biomarkers, if you will, it's still a very small field. The studies are really on limited samples, short study periods, very few longitudinal, not studying diverse populations, and there's very little replication. The technologies are all over the map. Passive wearables, apps, and so forth, used in all sorts of combinations and settings. So it's hard to compare them, and this gets to the standardization gaps and the fact that some of these are limited in their specification of the systems and the analytic algorithms applied, and there are issues of proprietary protection that get in the way sometimes. Usability and adoption is probably the biggest stumbling block in many ways to the further rational use of these technologies in trials and in research. And then finally, there are a number of deployment barriers, not least of which are the ease of use. It does require a certain amount of expertise. There are some costs, but perhaps most importantly is if you want things to be used, you have to show evidence of their efficacy and effectiveness. And so there's a lot of work needed to highlight a few of these areas of challenge. One area is usability that I mentioned. So this on the right is shown an interesting design site where you can take a known device or smartphone and place it in the standard pocket of a man or woman. These are obviously changing over time, but just to make the point that we assume that people will wear things or use things in a certain way, but often it's not considered at the front end of a project. People live in a real world. This is actually a couple that I'm conducting a video appointment with on their laptop. The couple are actually both were born deaf. the gentleman has or had Alzheimer's disease, and just kind of shows that to me the considerations about, well, what's really happening in the real world. Another related issue is sustained engagement. So if technology has to be tended to, or you have to attend to doing something, the engagement tradition has shown to drop off quite significantly. This is data from a recent review of Apple Research Kit studies across a number of disease conditions, asthma, sleep, Parkinson's. None of these were dementia, but the bottom line is the consistency of the data here is that despite enrolling thousands, tens of thousands of people initially in most of these studies, over 50% of the cohort ceased engagement after just less than 2 weeks. And then finally, just to make the comment that there's a huge cottage industry in digital cognitive tests in general, and I would make the argument that these are better in many ways because they are potentially more objective, and you can look at multiple meta aspects. But ultimately, in the end, if they're still episodically administered, they're not necessarily engaging. They generally are measuring only a single dimension or a few dimensions, and they're not ecologically valid. Nobody actually volunteers to remember lists of words or connect circles and dots. But these are of value, particularly probably I would say for screening more than anything else. So there's the now well-known digital clock drawing test, which gives interesting and added information. There's a whole host of these. This is some data from a colleague, Adri Seeley, using a touchscreen trails test, looking at different meta aspects of the test itself. But in general, as Henry Ford said, I think these are faster horses, not radical changes to the way that we could potentially get meaningful data. So what is the model of meaningful data? I would suggest the following, that our current legacy model is we identify people at risk. This is a photo that was sent to me by a daughter of a person who might have a dementia from their home. We see them in a clinic, and we do a mega assessment, and then we do some follow-up. Now, this has changed a little bit because we've become more adept or comfortable with televisits or televideo visits. But in the end, they're still episodic and brief. The way to potentially transform this system or this approach is to be able to take advantage of an array of sensors and devices that can provide information generally from the home or in the community across a range of important domains for health and wellness. And then integrating that information, obviously digitally, using the internet to provide data that's much more objective, continuous, real time, ideally ambiently, unobtrusively, that therefore makes it ecologically valid, and is a measure of really everyday function and cognition. It's really, cognition is really function. Every time you're thinking to do a task, a functional task, you're using cognition. And then finally, the other incredible opportunity here is that because of the high dimensional data, it really begins to allow one to look at intra-individual change, not just group changes or differences. So the way forward, I'm going to now turn to is, I think, trying to scale out and enable more people doing research to use these kinds of technologies. And this brings me to the CART initiative. The CART is an acronym, the Collaborating Research Using Technology Initiative, that was put forward by funding by the VA and the NIH, with the NIA taking the lead. And this was awarded to to OHSU. Actually, I want to give a shout out to Jason Karlawish, who was also a part of this effort, is a part of this effort. And basically, the call here was to create what I call an evidence engine for the wider use of digital methods in aging research, but importantly, to make sure that the technology was open, agnostic in the sense that it's not about a particular app, phone, operating system, that it's use case flexible and shareable. I think those are really some of the key points that had to be incorporated in this effort. And then ultimately, as I emphasized before, that we optimize for being unobtrusive and longitudinal over time. So what you see in this graphic is a bunch of domains of health and wellness. You don't see any technology because the technology shouldn't drive the function. It's the function that should drive what technologies are used. So building on a lot of work over the years through Orchitect, we have tuned that Orchitect platform into what's now the Orchitect Cart platform to create this technology agnostic, use case flexible, and shareable platform that takes the conventional assessments and research data as a given, but also allows one to add, if needed, EMR external data or even other sources of external data, such as weather or air quality, but to really focus on data coming from the home in these major domains of function and wellness. And then behind the function and wellness doors are any number of array of technologies that can be used and mixed and matched. So these are some of the signal ones that have been used most prominently. So just for an example, and I'm going to show more detail and examples in a moment, but so measuring cognition can be done with you can deliver a test direct to an individual on a phone, on a laptop, on a tablet, but you can also just look at how a person uses their computer day to day as a measure of their cognition. You can look at how a person remembers to take their medications using an electronic pill box as a measure of prospective memory. You can look at how a person drives, which is a measure of safety, but it's also a measure of cognitive function as well. So using a driving sensor shown on the lower right corner there. So there's an array of these technologies that are available. The system is always mindful of the fact that there are constant innovations and new sensors and methods, and it's been designed to incorporate those changes. Of course, the back-end part of this is extremely important. The data going to secure servers for analysis. I just put this up because I want to make sure that people realize that we all think. I mean, I'm guilty of this myself. I'm going to download an app and do something, and it's just so simple. Well, there's a lot of work that goes in the back end. I want to give a shout out to Thomas Riley, who's really the architect of all this system, who's just an amazing person. So let me just review a few of the domains and functions very quickly that have been studied and showed promise. So in the area of everyday cognition, computer use has been shown to clearly decline with people who have MCI over time compared to age-matched normals. So this is just here showing mean days on the computer, but also the number of computer sessions, the time of day. All of these are differentiating people with mild cognitive impairment from those who do not have mild cognitive impairment. Similarly, measures of mobility, such as walking speed, here showing the variability in walking speed over time, and even looking more closely, the differences in those with earlier versus later progression of mild cognitive impairment. Social engagement is a huge interest. It's incredibly important. And here again, Social engagement has been shown to differentiate people who may develop MCI or not. Interestingly, thinking about this as a caregiving metric as well is very attractive. So the time that people spend together or apart in caregiving is also available now through these kinds of methodologies. And then the final, just a small example, is sleep. So sleep can be measured a number of ways. It can be measured just with passive sensors in a room, particularly in those who sleep alone, but also with bed mats or with wearable. And all of these have been shown over time to differentiate people with mild cognitive impairment versus age appropriately matched normals. And even more interestingly, perhaps, is again being able to look at the interaction of couples. So there are differences in couples sleeping together, as many of us know, and these may also be important signals going forward. Now, all the data I presented before comes from passive sensors or wearables, or in the case of sleep, some bed mat data. But I want to emphasize that context is really important. And so we found that one needs to get this information. It's best gotten by asking people once a week through a pushed email, an old-fashioned email. It takes on average about 4.2 minutes to answer these key questions around these key domains. And we find that to have intelligent algorithms, at a minimum, you need this data. And it's very instructive. And just to show, this is some old, now old, it's amazing to talk about old COVID data. But just showing, these were individuals in the CART cohort. So one CART cohort is in the Portland low-income housing group, another is in Chicago in the Mars aging group. Just showing in the top panel changes in reported loneliness. The arrow is when the governor announced a state of emergency. I think that was in March in Oregon. And then in Illinois, in Chicago, showing this is activity data, but showing the effect of activity data in low versus high cognition individuals just based on cognitive test scores pre-state of emergency declared in Illinois compared to, and then going further forward. Validations are also very important. I'm just going to very quickly go through this. It's just to emphasize this point that there's a plethora of devices. Many of them need to be validated. There's some that are more research ready, research actigraphs, for example. These need to be validated with commercial devices because the commercial devices are not always known in terms of their internals or their proprietary algorithms. Similarly, things like everyday cognition with conventional tests, like walking speed, have been also cross-validated. There's also the idea of digital biologic validation. So, do these things relate to the actual underlying pathology? So in this case, this is a study by Lisa Silbert showing medial temporal lobe volumes are highly correlated with changes in computer use over time in people with MCI. And then even showing that in people who have been monitored at home in these various domains using the sensor system, that there's a correlation of both amyloid plaque scores as well as with Brock staging. So the last piece of this part of my presentation, I just want to emphasize that the importance of looking at different populations. So in CART, there were 4 specific populations. I mentioned the African American adults in Chicago. There was a Latinx population in Miami, rural residing veterans, and then low-income seniors in Portland. And the importance here is that they are diverse, even amongst the population. So, for example, there's some who are living alone, some are not, some are driving, some are not so much. Actually, in this cohort, many happen to be using a smartphone, but not so much iPhones. The ability to make sure that people would maintain the systems in their home or not want to withdraw was very important in this study. And that was, fortunately, good retention was achieved. And then just to show some detail about MCI. So this is just MCI divided by MoCA score. So a very simplistic way of looking at this for this purpose, but just showing that there was looking at a difference in MCI versus not in terms of wearing the wearable, but actually the compliance was different in the MCI group versus the matched controls. This was just a 4-month snapshot, but just to emphasize that even though compliance was high overall, there were these differences. There's a universe of people now who have been using this technology or parts of the platform, and I just want to take this opportunity for a shout out to Penn and colleagues there that hopefully we can work with you in that regard. And then finally, I just want to end with some comments about the promise in use in clinical trials. So this you'll hopefully remember is Ron Brookmeyer's table from the sample size estimates, showing how the way that we conventionally study people we need really high sample sizes. If you look at how digital biomarkers might actually improve the situation through simulations using real data, you can see here in this work done by Hiroko Dodge and then subsequently with Jiayi Wu, that just looking at the middle row, looking at an average effect size desired about 30%. Using conventional kind of measures, you need, not surprisingly, a couple thousand people, but using the digital measures, the sample sizes needed are drastically reduced. And this has actually been replicated in an independent sample. It's also been shown, for example, for sleep measures, total sleep time. And the principle behind this, if you will, is the conventional measures look like this. And you try to make some kind of estimate of a change based on that sparse data. You also get complications because the actual symptoms may be reported anywhere along the way, and you're trying to make sense of these relationships. If you have data all the time, you realize that there may not be a change, but there is a change, and the change is in the functional range or the variance, the data variance, and this may be a signal for early detection. And it really is the key to being able to look at intra-individual variability and intra-individual change, which really means that one can reduce the sample size. Importantly, the opposite is that means that you may be reducing the exposure potentially to harm. You can make more, as I mentioned, precise estimates of intra-individual change, and really, from an industry standpoint, inform go, no-go decisions much more intelligently. So this approach is being used in some trials. Right now, these are non-pharmacologic interventions. Hiroko Dodge's iConnect study, which is using social engagement enhancement by conducting daily video chats direct to home. This is just the design of the study. I'm not going to go into details. The study captures the voice as well as the video of these daily chats that are held as part of the protocol. The electronic med tracking is used to look at a functional outcome measure. There's some interesting data from the early the pilot studies that show, for example, that the audio that's captured can be analyzed offline, showing that, for example, in the conversations, there's a higher proportion of words spoken by people with MCI, and the content of the language, interestingly, may be different. So the graph just shows that there may be more swear words in the conversation. This is not people swearing like sailors. These are very subtle differences. Another study, which is being conducted by, led by Alison Lindauer, who's shown in the upper right on the video screen, is a caregiver intervention, but the caregiver intervention is being is for individuals who have difficult behaviors, dementia partner with difficult behaviors. Using the platform, Alison will be looking at, or is looking at, things like time out of bed, number of awakenings, time in the bathroom, out of the home, care partner activity as part of the outcome measures of the intervention itself. And then finally, I want to switch to the farther end of the spectrum, if you will, of dementia treatment, which is in desperate need of some kinds of help and treatments. And we've seen this in the COVID era that people in institutional settings have really struggled, and people with agitation, which Alzheimer's original patient struggled with, need help. And so a project that is being led by Michael Young is using the same platform. So this is the same technology, but just reconfigured to go into memory care units or at home for people with agitated behaviors. And what's shown here are some earlier results. This is looking at, so this is in memory care unit. This is actually just one patient to exemplify difference, looking at their movement about their room on agitated nights, which were actually marked specifically because the person received PRN medication compared to non-agitated nights. And the gestalt here, without going into details, is there are clear differences. Michael's gone on, though, to look at the environment by using sensing the light, the sound, the temperature, the humidity conditions in the rooms of the residents with agitation, and has shown that these all distinguish agitated nights from non-agitated nights, for example. So a lot of promise to use passive sensing as outcome measures or ways of mitigating disturbing behaviors for people with dementia. The future is bright, I think. There's so many other opportunities to look at going forward. We think that the idea of understanding a person's behavior, life cycle, their habits and patterns is really what changes as people develop MCI and dementia. This is a graphic from the Info We Trust site, just showing this is taken from diary data, I believe, constructing the daily activities of Darwin, who seemed to have some interesting activities. He took a tea with egg at somewhere around 10 o'clock or at night. But the idea, an old idea, is the same here. So this is a spiral plot of activities of a person who developed Parkinson's disease. Here shown when they didn't have Parkinson's. So these are 8 weeks going out over time, midnight, noon on the bottom. And you can see this is a wedge of less activity at night. The red is trips to the bathroom mostly. But then you see when the person developed Parkinson's disease, there was this quite disruptive behavior. You almost don't need statistics to look at that. And then this is with treatment with some Cinemat, so there's some normalization. And the same kinds of habits and patterns can be observed in MCI, shown here, just one person in a similar way. So going forward, there's a number of ways to look at this. We can just passively look at individuals' room transitions, or sometimes called also the opposites, dwell times. We can look at room occupancy on average over different time periods. And then these can be translated into what might be called life behavior fingerprints. And so Xiao Wu has shown that that people are, have these interesting patterns that are people with MCI are less likely to leave home in the morning, more likely to leave home in the afternoon, less likely to spend prolonged time in the kitchen or make trips to the kitchen in the evening, and so forth. And then finally, I just want to, so this is a climate change argument perhaps. So on the West Coast, in this past year, last September, we had terrible wildfires, which the fallout of which actually spread east in terms of air quality. This is just a map showing the colors are cycling over a 2-week period of these are the air quality where we had individuals in the CART study in the Pacific Northwest, and the darker the color to the dark purple is hazardous. But you can imagine now that there's increasing data suggesting that even air quality is a risk factor for Alzheimer's and the environment itself. We haven't had good ways of assessing this, and I think now we do. And so this is another kind of promising area. So with that, I realize time is short. I want to end and thank you so much for giving me this opportunity to at least come to Philadelphia virtually. I want to give another shout out to Collaborate. I really passionately want to work with others and see the value of bringing these kinds of tools and methodologies to wider use and sharing. And then, of course, I'm here for however long you want to ask questions or make comments. Thank you.
Thank you, Jeff. That was really a tour de force description of your work that I think has had so much influence on the field, and just the ability to start to link things like COVID and global warming, climate change with as factors, and to be able to study that in a quantitative way is truly extraordinary. What we're going to do is ask people to put questions in the chat, or you can unmute. But I think I'll start with one, which is, to some extent, to what degree have two questions. To what degree have you looked at the specificity of your measures with regard to different kinds of neurodegenerative conditions? You talked a lot about MCI and dementia, but are you also seeing signal that relates to the nature of the underlying pathology? And then somewhat related, but not completely, is to what extent are patient-related factors that you talked about, like education and for example, exposure to computers, influence outcomes, and how inert are these measures to those factors as well?
Yeah, so those are important and good questions. I think to the specificity to the disease, underlying disease processes, I actually, I mean no disrespect, but I actually think that that's not as important in the sense that, we use for example, the UPDRS has been used to measure aging changes, motor changes. It's not specific necessarily to Parkinson's disease. When you look at a gait speed can be slow because you have orthopedic problems or because you have Parkinson's disease. So really what's important is the context of use and knowing more about the group that's being studied. So I would suggest, as we now, I think, have learned almost the hard way, that in trials, you need to know what the target engagement is. So the biomarkers are incredibly important to say, if you think that the treatment is acting by mechanism X, do we know that this drug is really engaged to change that mechanism? And then the outcome measures are functional measures. It's, is this person's life improved? Is their ability to function improved? And that is in many ways, I think, irrespective of the biological mechanism. And then the second question about, the effect of different users is fundamental. First of all, the data we have about how people are using technology, particularly among the older population, is pretty much unknown. There are really two major sources of information, or two or three. One is the Pew American Life Survey. The other is the AARP survey of digital technology and use. And interestingly, the recent surveys that they've done actually have very different results. So one, I think the Pew survey suggested that something like 61% of people over 65 own a smartphone. The AARP survey was much higher, I think something in the 70s or so. So even at the sort of fundamental level, we have a lot to understand. We also, I think, have to be careful to not assume that people who are in low income or minority groups don't use technology. We found in the CART study that the low income housing group had a very high rate of smartphone use, and that was because they're used to moving. One of the reasons, and they actually do know that they can get some discounted plans, and it's not always. I think we have to be careful not to make assumptions about different groups. We need to make sure we document and know what these features are that might affect use, and that I think is the real key.
Great. I think that certainly makes a lot of sense. Others questions? I don't see anything in the chat right now, so if you want to just speak up, you're welcome to unmute and ask a question.
See, if we were in person, I just call on people.
You just, yeah.
No, I
well, I'll follow up with one other, and I think this gets maybe a little bit into specificity or at least signal to noise. How much have you looked within the kind of preclinical space, which has really, become obviously a target as well for disease and trials, and how sensitive are some of these types of measures to changes at that disease stage?
So the one of the fortunate things that we had happen is we started our installed life lab base with people who were not impaired, and then we were able to follow them for pretty long periods of time. And I will, let me just finish here, and I'd love to answer this next question. All of those areas that I showed, the mobility, gait speed, computer use, time out of home, sleep, those have all been shown to detect or predict incident MCI. So I think that there's good evidence that these are actually quite sensitive early markers of change. And I think we know clinically, when you see people in clinic with early MCI, people will say like, yeah, I still use my computer, but it's don't seem to remember the passwords maybe as frequently, or there's those sort of more subtle changes that I think are there to be picked up. There is a question, and I'll just read it. Most 80 study participants seem to be in urban and suburban areas. How applicable are these measures and technologies to those in rural environments? So that was a planted question, because it's a favorite topic of mine that we moan and groan about how, we don't have enough people to enroll in our clinical trials, but we exclude essentially because people have to travel great distances or may not have the transportation available, even actually more locally, because of where they live. So in the CART study, there was specifically a rural cohort. So these were people who lived hours away from any urban areas, that are classified as rural areas by this RUCA scale. And so once the technology is installed and turned on, it provides the data continuously without having to travel. I think with video now being available, this may help people who feel nervous about doing things entirely remotely without being able to put eyeballs onto, have an eye-to-eye connection with a person who's in a study. But I should also mention we've been able to successfully, driven by COVID, install the technology into homes entirely remotely. That is, without a person having to, from our technical team, go into a home. So we have a self-installation protocol now that I'll admit I was skeptical that this would work, but it's been remarkably so far successful. So I think that this is going to change things quite a bit. Being able to do remote installations and remote assessments across the board could really increase the number of people that we can enroll. Yeah, the other area that I am particularly concerned about is we require people to have a, quote, study partner. Now, the rules around what a study partner is are very loose. What does that really mean? And it's always been interesting that how strict trials are about, are the raters trained and certified? But we don't train and certify the study partners. We take anybody essentially who claims, yeah, I'm with this person 4 hours a week. Oh yeah, realizing that it's just to get the person in the study that people will say anything. So so that's a big problem, and I think actually huge number, over a third or more of people 65 live alone, and that number is growing. And as you get to older groups, the groups that's most at risk for Alzheimer's disease, 75, 80, the numbers are, turn to be 60%, particularly among women, live alone. And so we're shooting ourselves in the foot again in terms of being inclusive in clinical trials. So that's maybe a bit of an editorial kind of comment, but.
Well, I certainly
somewhat negative opinion.
Oh, there he is. John, you're trying to speak?
Yeah, it sounds like a somewhat negative opinion. It's like, will we ever be able to advance with clinical trials? There being so many variables and so many differences that it's hard to see movement in one direction or another. Are we forever lost in the confusion of behavioral confounds?
Well, actually, I hope I haven't given a negative. In fact, if anything, I'm very excited and positive about our prospects because we can change what we've been doing. When my title of my talk is Changing the Way We See Alzheimer's. I think that we've been stuck in using the same approach for too long, and actually we have some new approaches that can really change things. And there's something that we can, even with high dimensional data, you can do valid N of 1 studies. We all talk about personalized medicine or precision medicine. We haven't really addressed this appropriately on the clinical outcome side, when in fact there's well-known methodology, accepted methodology to do true N of 1 studies that would allow us to deal with the heterogeneity of Alzheimer's disease in a trial setting, in fact. So I'm actually quite positive. I just think we need to see more.
Name me one advance that has advanced Alzheimer's clinical trial in this population of population of people, one facet of their behavior that has definitely improved in 90% or 80% or something. um, be it being able to go shopping or tying shoes, managing clothes. What are the things that we're seeing that have improved?
Well, that gets to, it's an interesting question, John. It's a conundrum that um, in part in Alzheimer's disease, we haven't yet seen a treatment that moves the needle appreciably. You could argue, for example, that the cholinesterase inhibitors, when you look at group data, are pretty meh. But you know, the number needed to treat to see an efficacy for a cholinesterase inhibitor is about 12 patients. So there is perhaps a subset of patients who actually do have a pretty noticeable effect. And clinically, you know, we believe we see this. You know, you have to be careful. The plural of data is not an anecdote. But I think that those of us who have seen more than 12 Alzheimer patients treated with these drugs will see patients who seem to genuinely have an effect. In fact, they go off the med, and which is actually kind of an N of one experiment. They go off the med, they get worse, they go back on, they seem to actually get better. So I think that we can see these effects, but you know, one of the things that I think we've been struggling with a little bit in the field is that with the history of you know, taking the ADAS-Cog and reconfiguring it to the PAC and the IDRIS and all these different composite measures is we sort of keep using the same measures and saying it's just the measures perhaps that aren't working, when in fact it's the drugs that may not be working so well.
Sure.
So it's hard.
So do you think we'll get to a penicillin that has the same effect on polio that that penicillin had, or will we always be chasing our tail trying to find a better therapy without ever really succeeding in doing something substantive?
Well, I believe we will. But that's easy for me to say. It's like being an astrophysicist. You know, can you do the experiment now that will show it in the future? I do think that so again, this is easy to say now, but speculation. But I think that there are, and we're seeing this truly, that there are there are subtypes or groups of individuals who have different pathophysiologic drivers of the disease, and that we will, just by dumb luck, in fact, I mean, many of the drugs that we've discovered were serendipitous. So, you know, the multiple mechanisms now that are being addressed in trials, I suspect that we will see some movement in the next decade. I really do think that's,
okay.
Yeah.
Okay.
There's another question from the chat. I can read it to you if you'd like. The longitudinal effect sizes you report are incredibly impressive. And I'll preface the rest of the question by that Paul Yushkovich, who asked it, is an imaging person and expressing his anxiety. Do you see the role of expensive imaging and invasive outcome measures recede in clinical trials as these measures emerge? And do you envision using these measures much earlier along the disease spectrum to predict progression in MCI and dementia?
So that's a great question. So I cut my teeth on imaging. I used to be deeply immersed in image analysis research. I actually chaired the neuroimaging PIA session probably about 15 years ago. I was very immersed in that area, but I switched to the digital biomarkers because I saw that there was no pathway for the imaging group. No, totally not the case at all. In fact, I think, you know, I have a slide that I chose not to show, but I think that really the important role of imaging is is the idea of identifying the targets and knowing that the treatments are actually engaged in what you think is the pathophysiology of the disease. And so I think that's number one. And number two, related to that, is identifying people at risk. It's sort of it's a trait. They're trait markers that are really important. And so I see this as, I see the imaging in particular, and the blood-based, the fluid biomarkers still playing a huge role, particularly in screening, in validating targets, engagement, and efficacy of the drugs in that way, in concert with actually seeing whether the clinical outcomes that we desire are actually, proceeding. If we had large amounts of unlimited funds, you could argue, well, we'll just look at clinical outcomes, and who cares about the mechanism? You could do that, but I think that's incredibly inefficient and just adds to the confusion in a way. So I certainly don't advocate throwing out the other biomarkers in trials at all.
That'll make many of us relaxed moving forward. As we've gone over in time, I'm just going to ask one other final question, which is, data is not inexpensive, right? And you're collecting a lot of it. Have you looked at how much you actually need to collect of this data? Like, do you need computer use data every day, or can you reduce that to once a week, or or have you thought a little bit about sparing server and storage costs in that regard?
Great question. So it's kind of there's several pieces to the answer, I think. One is the sampling frame question. So you can't see a difference if the human response is not in that frame of reference. So a simple example is if you're looking at reaction times and your technology doesn't capture beyond a second of data at a time, then that doesn't make sense to capture different time lengths or spans. But the data, the majority of this data is, in terms of actual capture and size, is surprisingly small in today's world. The big data is really the video and audio files. So in terms of actual, how those can accumulate quite quickly, and so in terms of the size, and big data has the different dimensions. So I think we often get concerned about the size, and in many ways, I think the size is the least of the problem. Storage is relatively cheap. It's processing that becomes expensive. So if you want to take these large data sets and move them out of the cloud and back in, and so that's where cloud services get you. And so those costs currently can add up quite quickly. But the collection of the data, if you're looking at computer use, once things are set up, electrons are cheap. So there's no harm in collecting, in some ways, too much data. And in fact, the beauty of that is once it's collected, you can go back and reparse it and look at it again. And then, years later, in fact, you can go back and look at the data. We just looked at some old data from years ago of people with landline phones. Remember those? Some people still have them, and that's okay. But And we were looking at landline, we were looking at phone use in older people who had only landline phones. And that, that was data from 15 years ago, I think. And we were able to show, because we now had seen this group of people they were all normal time, but they subsequently, a decade later, a number of them, a considerable number, had actually developed dementia. So we could then go back and look at those sort of electronic records of phone use in this case, and show, very interestingly, that their phone use was clearly different. And interesting, the callers to and from, they're sort of, going back to the social networks, there's sort of this contraction of their social network as what appeared to be, in retrospect, their development of cognitive impairment progressed.
Very interesting. And yeah, I was motioning to someone to hang up their phone the other day, and I realized they didn't know what that motion was anymore. So, well, I think I can speak on behalf of everyone that was a truly wonderful talk and very engaging discussion. So we really appreciate, again, your electronic travel here today to Penn, and hopefully we can get you here in person in the future, and maybe even think about some ideas for collaboration moving forward.
I'd very much enjoy that. That would be terrific. So thank you so much.
Thanks, everyone.
Great, thank you.
All right. Be well.
You too.
Bye.